Cellular Interactions, Part 2. In the first part of this topic, we talked about three major types of junctions that are formed between cells: occluding junctions, anchoring junctions, and gap junctions. In Part 2, we are going to talk about some of the molecular mechanism that form these junctions and some of the important proteins in these junctions. So the cell interact with each other and with the matrix through these transmembrane linker proteins. And when a transmembrane linker protein binds to an identical transmembrane linker protein on a neighboring cell, this is known as a homophilic binding. When a transmembrane linker protein binds to a different protein in a neighboring cell, this is known as a heterophilic binding. And of course, the interaction of a cell with an extracellular matrix component is always considered heterophilic binding because the receptor is binding to something different in the extracellular space, for example, an extracellular matrix protein. Sometimes the cell can interact with each other through an extracellular linker molecule, and this is shown in the right side of this slide, but this is very rare, and we are not going to consider these types of interactions. We're just going to talk about the homophilic interactions between the cell and the heterophilic interactions between the cells and the cell and the matrix. So this slide shows two examples of the homophilic interactions and two examples of heterophilic interaction. So for an example, the transmembrane linker protein cadherin participates in homophilic interactions with cadherins on a neighboring cell. There's another family of cell adhesion molecules, such as an immunoglobulin superfamily of cell adhesion molecules, or CAMs. An example of this is NCAM, or neural CAM, where in the nervous system that is found, and it participates in homophilic interaction with CAMs on neighboring cell. There is also evidence shown on the far right side that the Ig superfamily cell adhesion molecules can interact with integrins on neighboring cell, and this would be considered as heterophilic interactions. The transmembrane linker protein integrin can interact with the components of the extracellular matrix. This would be a heterophilic interaction. And finally, a transmembrane linker protein known as selectin, an example is P-selectin, can interact with a glycoprotein of a neighboring cell. And this last heterophilic interaction, you can see that really important in the white blood cell function. I want to look at each of these proteins and each of these interactions. So let's start with cadherin. Cadherins are a very, very large family of cell adhesion molecules. You will remember they represent the transmembrane linker protein in adherence junction as well as desmosomes. The first three cadherins to be discovered are shown in the table. You have E-cadherin on epithelial cadherin, which plays an important role in junctions in embryos and non-neural epithelial tissues. Then there is P-cadherin or placental cadherin that plays also an important role during development, and you find a high abundance of this transmembrane linker protein in placental epithelial cells and also in skin epithelial layers of the epidermis. And then there is this N-cadherin or neural cadherin, which play a role in junctional complexes in nerve cell, in lens cell of the eyes, in cardiac muscle, and skeletal muscle. And you remember we talked about several of these cadherins as well. Desmoglein and desmocholine are members of this family, and they are the transmembrane linker proteins in desmosomes. So this is a general structure of cadherin. They are glycoproteins that are made up of between 700 to 750 amino acids. They have a very interesting 5 domains in the extracellular space shown here, and each of these domains is about 100 amino acids. And 3 or 4 of these domains are often able to bind calcium. And so the binding of calcium is really, really important in the function of cadherins. Then the cadherin has an alpha helical transmembrane region, and then a C-terminal region can interact with a variety of plaque proteins, in this example, catenins and vinculin, which can connect the cadherin to the actin. And of course, when the cadherin is bound to these plaque proteins as well as cytoskeletal filament, this is referred to as a junction. So this is a diagram that shows some cadherins with a little bit more molecular detail of other proteins involved in a typical adherence junction. So you'll notice that the cadherins, which are light or dark purple right here in the center, are in pairs or in dimers. And so E-cadherin forms dimers with other E-cadherins on the plasma membrane, and then these dimers on one cell can then interact with the dimers on the neighboring cell. So you can visualize the plaque proteins here as well. So in one cell, so this is a cell on the left, has the beta-catenin, then alpha-catenin, and then ZO1, which can connect the cadherin to the F-actin. And the cell on the right is using different types of plaque proteins: vinculin, VASP, alpha-actinin, and P120-catenin, connecting the cadherins to the actin filaments. This diagram is just an illustration in terms of plaque proteins. The plaque proteins aren't necessarily different in neighboring cell. So in this example, here's one cell is using one set of plaque proteins and the other cell using a different set of plaque proteins. But that's not always the case. Sometimes neighboring cells are using the same type of plaque proteins to connect the cadherins to the F-actin. So cadherins form homophilic interaction with cadherins in the neighboring cell. And this is really well illustrated in an experiment that was done quite a few years ago, and it is shown in this cartoon. There is a type of cell called an L cell, and these are found in the intestine. A mouse L cell do not normally express cadherin. So you can genetically engineer mouse cell to exogenously express cadherin. So in this experiment, the researchers engineered mouse L cells to express E-cadherin, P-cadherin, and N-cadherin. And when all these cells were mixed, and what they noticed that the cells expressing E-cadherin will only interact with E-cadherin. Cells expressing P-cadherin will only interact with the P-cadherin, and the cells expressing N-cadherin will only interact with N-cadherin. This experiment was a very clear illustration of how cadherins form these homophilic interactions. And this is a series of images, which is a time-lapse video fluorescence microscopy of cells that are expressing GFP-tagged cadherin. And you can see that when you mix these cells, after about 8 hours, these 3 cells are interacting very tightly, and you can see a strong accumulation of fluorescence and therefore the strong accumulation of cadherin between the cells. So cadherin is really important for connecting cells, and again it forms a calcium-dependent homophilic interaction, and that's pretty much all about cadherin. So now let's take a look at example of an homophilic interaction, another example, and that is the Ig superfamily of cell adhesion molecules. This is a very, very large family of cell adhesion molecules, hence the name superfamily. And they are called immunoglobulin superfamily cell adhesion molecule, not because they are immunoglobulins, not because they are antibodies, but they have a series of extracellular domains that are very homologous to the domains that you see in the antibodies. But they aren't antibodies, and they can form calcium-independent homophilic interactions. So Ig superfamily CAMs on one cell will interact with the Ig superfamily CAMs on the neighboring cell, and they can also form heterophilic interactions with integrin. And again, this is a very, very large family of cell adhesion molecules. In nerve cells, these are called NCAMs or neural CAMs. In endothelial cells, they are called ICAMs, and they can take on all different kinds of membrane topology. Some CAMs, some of the Ig superfamily CAMs shown here, are transmembrane proteins with very large C-terminal cytoplasmic domain. Next to that is a transmembrane Ig superfamily CAM, which has a very short cytoplasmic domain. Some of these Ig superfamily can interact with the membrane through a GPI anchor, and actually some of these are released in the extracellular matrix space and actually become the part of the extracellular matrix. So now let's go on and look at the next type of interaction. So in this case, it is a heterophilic interactions, and that is the interaction of the integrins shown here with the components of extracellular matrix. So at the beginning part of our discussion on cellular interactions, I told you that the cells interact with each other in tissues and organs, and they also interact with the components of the extracellular matrix. And the protein responsible for that is integrin. Now, integrins are a very large family of proteins, but these proteins all have the same structures. They all have two subunits, an alpha subunit, shown on the left in the green, which is again a transmembrane protein. The alpha subunit or the alpha chain is actually in two parts that are connected by a disulfide bridge. The alpha subunit is associated with a beta chain, which is also the transmembrane protein. The beta chain has a characteristically cysteine-rich region in the extracellular space, and the C terminus of the beta chain can bind or interact with the proteins of the cell to connect the integrin to some type of cytoskeletal filament, such as actin or intermediate filament. On the alpha chain, there are 4 binding sites of calcium or magnesium. This is really important for the function of integrin. It is calcium-dependent binding. So in other words, calcium has to be bound for the alpha subunit to be in the correct conformation to function in cell matrix interactions. And I mentioned that the integrins are very, very large family of proteins, and there's probably more than 22 integrins that have been described. And what is interesting is that just by swapping out the alpha subunit or the beta subunit, you can actually change the binding specificity of these cell adhesion molecules. So in this table, you can easily see an integrin that is made up of alpha 1 or beta 1 subunit will bind mostly to the collagen in the extracellular matrix. However, an integrin that is made up of alpha 2 subunits and the beta 1 subunit will bind mostly to the collagen, but it also has the capability of binding to laminin and so on. So other example of the various integrins and how the subunit composition changes the ligand that it binds. This is just to illustrate that you can have multiple permutation and combinations of the alpha and the beta subunit to alter the specificity. So really interesting is that the cells can regulate the activity of integrin. So they can activate integrin and they can deactivate integrins. So in the top part is an illustration on how integrins become activated. So what you have is in the plasma membrane, in the green is an inactive integrin, and next to it is a receptor for cellular signaling. And then you have an extracellular space in yellow. You can see that some components of the extracellular matrix, something that the integrin can bind to. You have a signaling molecule that comes in and binds to the receptor. This activates the receptor. And then through a series of relay proteins, the integrin can be activated. And when it is activated, it becomes adhesion competent and then can bind to the extracellular component. Cells also can turn off their integrins by phosphorylating them, and this is shown in the second part of this slide, or the section B. So you see a couple of integrins. They are bound to the components of the extracellular matrix. You can see the intracellular attachment proteins that have attached the integrin to the cytoskeleton, to the actin cytoskeleton in this example. Now, kinases within the cell can phosphorylate the serine residues on beta chains, and this causes the entire complex to fall apart. And in doing so, you have deactivated the actin. And this is really important for a number of reasons. One of them we talked about already. Remember, we talked about motile cells. When the cells are moving, they have continuously form or produce or generate and break the focal contacts. because as the cell is moving along a substratum, focal contacts are being made at the leading edge of the cell, and the focal contacts are being broken at the trailing edge of the cell. And so this is the way which the cells can do that, by regulating the activity of the integrins. Now, regulating the activity of integrins is also important during mitosis. You can imagine that when a cell is dividing and the two daughter cells are pulling apart, you don't want these daughter cells to be connected too tightly to the neighboring cells, or they won't be able to come apart. And so during mitosis, cells often downregulate the activity of the integrins to facilitate the cytokinesis of the two daughter cells. And interestingly, in malignant tumor cells, often abnormally downregulate the activity of integrins. And this makes sense because tumor cells are very rapidly dividing cells. They're undergoing abnormally high levels of mitosis, so active integrins would be detrimental in that case. And also, inactivated integrins also facilitate metastasis or spreading of these tumor cells. So this figure shows a model of integrin activation. And it is thought that integrin activation comes about by significant changes in their conformation. So in this inactive state, the alpha and the beta subunit are kind of bent over onto the membrane. But when the integrin becomes activated through the cellular signaling, the alpha and beta subunit, well, you can almost think of them are standing up, and they become adhesion competent and can interact with the components of the extracellular matrix. And then another interesting aspect of integrin is not only are they activated by cellular signaling, but they themselves can serve as signaling molecules. So their only job is not just to bind to extracellular matrix and connect cells through cytoskeleton, but they can also propagate signals. And this slide illustrates some of the signaling pathways that can be propagated from integrins. One of these pathways we have seen already, and it is the pathway that is also propagated from the receptor tyrosine kinases. So on the right-hand side of the diagram, you can see a dimerized active receptor tyrosine kinase. It is bound to its ligand, so the receptor can activate GRB2, which in turn recruits SOS, which is the guanine exchange factor for RAS. RAS in turn activates RAF kinase. RAF kinase can then activate the MEK kinase, and then MEK kinase can activate the MAP kinase, which then go to influence the transcription. Interestingly, integrins can activate the same pathway, and integrins can also activate other pathways as shown right here. So we have looked at the cadherins and the immunoglobulin superfamily cell adhesion molecules, which fall under the homophilic interactions. And we looked at the interactions between the integrins and the fibronectin, which falls under the category of heterophilic interaction. Now, on this slide, I want to look at a couple of other interactions, and that is the interaction of the P-selectin with glycoproteins on neighboring cells, and the interaction of immunosuperfamily CAMs with integrins. Both of these are important in white blood cell function. So in this diagram, what you have are endothelial cells here in the beige, which line the blood vessel. And on the surface of these endothelial cells are the ICAM1 and ICAM2. These are the members of the immunoglobulin superfamily of cell adhesion molecules. And within the endothelial cells, there are vesicles that contain another adhesion molecule, P-selectin. Now, within the bloodstream, you have the white blood cells or the leukocyte, and they are shown here in the blue, and they have a number of interesting molecules on their cell surface. First of all, they have what is known as a mucin-like CAM in yellow, labeled to as selectin ligand, and it is the ligand for P-selectin. And you also have the integrins on the surface of the white blood cells. And in this example, it is an alpha L beta 2 integrin, which can interact with ICAM1. And finally, you have a signaling receptor known as a PAF receptor in gray, which stands for platelet activating factor. So in absence of any signal, the white blood cell will flow through the blood vessels and it will not interact with this endothelial cells. And it will just flow along with the movement of blood at a very fast pace. However, if there is some kind of an infection or an injury to the tissue, you have an inflammatory response. An inflammatory response is the secretion of chemokines and cytokines from the cells at the site of the tissue injury or from the site of infection by a pathogen. These chemokine cytokines are small signaling molecules and act upon the endothelial cells of the nearby blood capillaries in the tissue where the injury or infection occurred. These signals will cause an activation of the endothelial cells that line the blood vessels such that the P-selectin is now trafficked to the cell surface. And you get also the synthesis of a cell surface signaling molecule, platelet activation factor, PAF. When this happens, these endothelial cells are activated, and as the white blood cell flows past the P-selectin on the surface of the endothelial cells, the white blood cells will be trapped through the interaction within the mucin-like CAM, which is the glycoprotein on the surface of the white blood cell. So you end up with the white blood cell being trapped by the endothelial cells. This interaction caused the leukocyte to roll over the endothelial cells. They're not flowing past the endothelial cells in a fast pace as before, but now rolling slowly on them. They are still rolling slowly due to the shear force of the blood flow and a relatively low affinity interaction between the mucin-like CAM and P-selectin. They're trying to hold onto the P-selectin, but as these interactions are sufficiently low affinity, they are forming and breaking due to the shear force of the blood. They cause the leukocyte to roll along the endothelial cells. Then what happens as the leukocytes are rolling in this third step right over here, the platelet activation factor receptor on the white blood cell will interact with the PAF on the surface of the endothelial cell, and this will activate the integrin. The integrin will become adhesion competent and will begin to interact with the immunoglobulin superfamily cell adhesion molecule, ICAM1 or ICAM2, on the endothelial cell. This will bring the white blood cell very, very close to the endothelial cells. Now you can think that this interaction is high affinity or trapping with a stronger force anchor compared to a weaker affinity or force or anchor by P-selectin alone when the leukocytes were rolling. And then the activation of the integrin can also activate the motility of the white blood cells, and that white blood cell can actually crawl through the spaces between the endothelial cells and move into the site of infection or move at the site of the injury as a part of the inflammatory response. So there you can see a combination of different cellular interactions. This crawling out is also known as diapedesis in physiology. Interaction between integrin and the Ig superfamily CAM, interaction of the P-selectin and the glycoprotein mucin-like CAM, as well as signaling through a cell surface receptor PAF. All three of these functions come together to draw the white blood cell out of the bloodstream to the site of infections, where they needed to fight the infection. So this final slide is just a summary of all of the junctions that we talked about. We talked about the tight junctions, shown here close to the apical side of the epithelial layer. Then there are adhesion belts right below these tight junctions. There are the desmosomes right below those adhesion belts. And then below desmosomes are the gap junctions, and all of these are cell-to-cell interactions. At the bottom of this figure are the hemidesmosomes, which are integrin-based cell matrix junctions. And there are focal contacts, which are also integrin-based cell matrix junctions. In the case of hemidesmosomes, the cytoskeletal filament is the intermediate filament, and in case of the focal contacts, the cytoskeletal filament is actin. And then on the right side of this image are some specific protein interactions that we looked at. So if you look at the bottom, the interaction between the integrins and the matrix, and then you have interactions of the cadherins with each other, interaction of the immunoglobulin superfamily of cell adhesion molecules with each other, the interaction of the same family with integrins, and then you have the interaction of the selectin with glycoproteins known as the mucin-like camps. And this is just a slide that summarizes all of the junctions. This concludes our discussion on cellular interactions.